Hi guys, today we're going to go over A1.2 nucleic acids, but we're only going to cover the SL portion. For HL students, you can head on to another video after this one. Basically, in this topic, we're going to learn about two genetic materials or molecules, DNA and RNA. We have to know the full forms of these. DNA is deoxyribonucleic acid and RNA is ribonucleic acid. D for deoxyribo, R for ribo. This is to do with the differences in their sugars. We're going to get into that later. But basically, what we need to know is that these two molecules store genetic information long term for all living organisms on Earth. However, sometimes RNA or DNA, well, all the time, it's either RNA or DNA that's found in viruses. But one key thing to note is that viruses are not considered living. And that's because they don't go through all the processes of life, which, for example, they can't reproduce by themselves. They require a host to do so. They have to infect a host in order to reproduce, and therefore are not considered living. So DNA and RNA are found in all living things, living organisms. They store genetic information, but also in viruses. DNA and RNA are polymers made up of nucleotide monomers. So there's a repeating chain of nucleotides. The components of the nucleotides are as follow: phosphate group, a pentose sugar, and a nitrogenous base. We have to know what these structures look like for the IB course. The pentose sugar just means they have five sugars, five carbons, sorry. One, two, three, four, and a fifth one up here. As I mentioned before, DNA and RNA have different sugars, but we're going to get into that later. There's also a phosphate group. A key thing to note about this is that it's what makes DNA acidic, and it has a negative charge. And the nitrogenous bases are what actually make up the DNA sequence or the base sequence, which allows us to store that information. Um, However, how are these nucleotides actually joined together? They're joined by something called a sugar phosphate backbone. So if I just copy multiple of these nucleotides together, they're joined so like this. Well, this is a bit low. The pentose sugar connects to the phosphate, creating this kind of zigzag pattern, which is what we call the sugar phosphate bone. Okay? Um, and this is when a covalent bond, so these are covalent bonds that form between, again, the sugar and the phosphate. These covalent bonds specifically, though, are called phosphodiester bonds. Keep in mind, other components also have covalent bonds. Like this is a covalent bond, this is a covalent bond, this is a covalent bond. These are covalent bonds. But phosphodiester bonds are specifically those that join one phosphate group of one nucleotide to the pentose sugar group of, or the pentose sugar of another nucleotide. Now, because there's so many of these covalent bonds, it requires a lot of energy to break. And because of this, it gives the DNA double helix, the DNA structure, strength and stability. Well, one more thing to keep note is that phosphodiester bonds, or the connection between these two nucleotides, are formed by something called condensation reactions. We're going to get into these reactions in another video. Now we're going to go over into more detail about the specific bases, these nitrogenous bases, and how they form the code of genetic information. Let's go over the specific details. So as we know, well, there's RNA and there's DNA. And these are the bases they have. RNA has uracil, adenine, cytosine, and guanine. DNA has all of these, but except instead of uracil, it has something called thymine. So, um, Yeah, A or adenine binds with T, and C binds with G. Adenine binds with U as well, in RNA specifically. However, so what we need to know though is that A to D has two hydrogen bonds, and C to G has three hydrogen bonds. And this is how I remember. This might not apply, like might not work for everyone, but this is how I remember. So A and T, if we know A and T bind together, T is the beginning of two, so that therefore it has two hydrogen bonds. C and G, if you stack them over each other, they look like a backwards three, so they have to have three hydrogen bonds. Or like C and G are very curly letters. Like the number three is curly, so it has hydrogen bonds, three hydrogen bonds. That might not work for everyone, but it does for me. It's these sequences of bases that stores information in RNA or DNA. So if we imagine this earlier on DNA polymer or RNA polymer, it would be like, let's say that's the sugar phosphate backbone. It would be A, T, A, C, G. That's one DNA strand. It's these bases that form that genetic code. Okay? Just to put on the record, RNA also goes through the same condensation reactions as DNA, but it is single-stranded while DNA is double-stranded. And it looks exactly the same. This could also be an RNA strand. Yeah. But let's go more into depth of the DNA double helix. We must be familiar with this sort of structure of DNA. You know, the two little strands, but I've only drawn a single strand. I've only drawn that one strand. How does it actually look like? So, again, DNA is this double helix. It's made up of two strands. That's why it's a double helix. So they're two strands. Yeah, these two strands are joined together by hydrogen bonds. So if I drew this one nucleotide and another nucleotide, this is the second strand, this is one strand, this is another strand. The hydrogen bonds here are what keeps them together in this structure to make it a double helix. Okay, but one thing we have to keep track of with DNA is that it's anti-parallel. That means they do go parallel, the two strands are parallel, but one is flipped over to the other side. Let me try draw this upside down to make it make more sense. But essentially, one is just flipped the other way. They both are built the same, but they're just flipped in the other direction. So while they're parallel, they both go straight next to each other. One of them is just in the opposite direction. SL, this is all you need to know. They go in a 5 to 3 direction. 5 to 3. And the other one would go 3 to 5. But we're gonna go more in depth in this for HL students in the other video. I mentioned so many similarities and differences between RNA and DNA. Let's like fully write them down. Venn diagram, RNA, DNA. They're both polymers. They both have nucleotides, and these nucleotides have the sugars, the phosphates, the bases. However, there's some differences. So DNA has deoxyribose sugar, as I mentioned before. RNA has ribose. Okay? DNA is double-stranded. RNA only has one strand. RNA oops, sorry. RNA has uracil, but DNA has thymine instead of uracil, but they all have adenine, cytosine, and guanine as their other things. For this spec, we do have to know some examples of nucleic acids, but I'll let that freedom be up to you guys to figure out what you want to know. Let's go more in depth about the complementary base pairing. This is a term we're going to hear a lot in this topic and in other topics. But it's what allows genetic information to be replicated and expressed and whatnot. It's based on hydrogen bonding. So let me just draw these two again. I know I've already stated it, but The only way a DNA knows what other nucleotides to put in the opposite strand is because of this complementary base pairing. If it was A, it has to be T. If it was T, it has to be A, it has to be G, it has to be C, it has to be G. It has to be complementary. And that's what allows for the stability, high accuracy in a lot of processes. It's just really necessary to keep the double helix in place. And these hydrogen bonds, for example, A couldn't bind with G because A requires two hydrogen bonds, but G requires three, and there'd be some kind of a mismatch. Okay? Um, and these, like I said, are very important for processes that occur within our body. For example, DNA replication. It's important for protein synthesis, again, within our body, like transcription and translation. And it's because of the hydrogen bonding and these complementary base pairs that allows for a high degree of accuracy in DNA replication, in protein synthesis, and all of that. And if we didn't have this, we wouldn't have functional proteins that we have. We wouldn't be able to undergo mitosis and meiosis to grow and repair cells and reproduce. So that's why complementary base pairing is so important. Now this section is going to be a bit mathsy, so bear with me. DNA is very um is very diverse. There's a lot of variety within DNA, and that's because each strand can have any length and any sequence. Like by that, I mean it can be as many as 3 bases to 1,000 bases, and any sequence it can be ATTCG or AAA TTTGGCCC. It can be any sequence and any length, and that's what allows for so much variation within DNA. So we have 4 bases, right? In DNA specifically, A, T, G, and C. Okay? Now, if we had a chain of only one base, it could be either of the four. That's 4 options. If we had a second base, it could also be either of the four. Now, this allows for 16 possibilities, and that's only with the two base chain, right? So every strand of DNA essentially has 4 to the power of n. Now, you don't need to know this formula, but it is really helpful for explanations and extended response questions, or just in general for your understanding. Okay, now going back to how I said genetic information is stored in these sequences, every base of 3 is contains like useful information. So if we have 3 bases, any of these bases, let's say 4, 4, 4. This is what you call a triplet codon. And each triplet codon codes for a specific amino acid, which then allows for proteins to be synthesized either as structural proteins or functional proteins, whatever. This triplet codon is what contains that information that we need. And there are many triplet codons. So for each triplet codon, there are 64 possibilities. 4 times 4 times 4. 64 possibilities for every triplet codon. Again, showing that variation. Um, yeah, that's pretty much it. Now, this is a bit brief, but we have to know that DNA and RNA are in all living organisms and viruses. But all of us come from the same genetic code. And we have this evidence from something called LUCA, the last universal common ancestor. Sorry for the spelling. Um, and we get into this in the topic about origin of like organisms and species and all of that. But this basically means that the very last organism that had same genetic code, it's the most common, the earliest ancestor that has the same genetic code as all of us is what we call LUCA. And that is evidence for the fact that genetic code is passed along and it's conserved and it's in all living organisms. And we all split from the same branch from that LUCA. I know it's a bit hard to wrap your head around if you don't have the background information, but we will get to it in another video. But basically, the whole point of that is that the conservation of genetic code is across all life forms, and we know that because of LUCA. And that's pretty much it for the SL portion. I'll see you guys in the other video. Thank you.